central tendons, uh, central tendons, central tendons to central tendons to the uh, the central tendons to down top the diaphragm. Three borders. Right border ventricles are the formed the entirely by the right atrium. Left border round is mainly formed by the left ventricles and partially by the left article. The inferior borders horizontal and formed by the right ventricles and cardiac apexes. So the four grooves, corny success sulks and circular sulks, which makes which marks the division between atria and ventricles, contains the trunks of the coronary vessels and completely ventricles that heart. The interior groove, the separate the two atria and is hidden by a pulmonary trunk and aorta in front. The intraventricles grooves, anterior and posterior, mark the division between ventricles which separate the RV from the LV. The two grooves extended from the bases of the ventricular position of the notch called is the cardiac apical inclusor. Then this is the covering of the heart. The heart is a pericardium, a double-walled sac around the heart, composed of a spherical, uh, superficial fibrous pericardium and deep two-layer serous pericardium. The the partial layer lines the i- internal surface of the fibrous pericardium. The vesicular layer of the epicardium lines the surface of the heart. They are separated by the fluid filled with the pericardial cavity called with the pericardial cavity. The pericardial cavity is the functions is mainly two things: protects and anchors the heart, the prevents the overfilling of the heart of the blood, the allows for the heart to work in a reactively friction free environment. Then the next one, internal spectrum. See, this is the anatomy structure of heart. The internal spectrum, located between the right and left arteria, contains fossa valis. The intraventricular spectrum, located between the right and left ventricles, upper membranes part, thick lower molecular part. Then this is a hard to dissection. So complete four chambers here. Ventriculars. This is a left to articulus. So four chambers here. The bundle of is here. So this the structure of then pericardial layer, the pericardium, myocardium. So this is the covering to the heart. This is the layer of pericardium layer. This is the myocardium. The heart is continuation process. It is the involuntary muscles activation. Involuntary muscle activation. Then next one, the layers of the heart wall. The heart wall layers, epicardium, vascular pericardium, then myocardium, cardiac muscles layer, forming the bulk of the heart. The endocardium, endothelial layer of the intramyocardial surface. So this is the X-ray image of normal and LVH, left ventricular hypertrophy. So the portion section, the complete labelings of art and the structures. This is a very important of diagram for the understanding to the how to functioning of heart and and blood circulations. Then artery of the heart is a very important artery of the four chambers, two chamber. Artery receiving the chambers for the heart. The receive venous blood returning to the heart, separated by an internal spectrum valve. The foramen valve opening in the internal spectrum in the fetus. The fossa ovalis remained from the foramen ovale. Each atrium has a producing auricle. The pectinent muscles mark arterial walls. The pump blood into the ventricles. The blood enters right artery from superior and inferior venu can and coronary sinus. The blood enters left artery from the pulmonary venous. The next is this is the diagram. The next slide is the left auricles, the projecting to the right pectating muscles in the wall. The four lines orifices of the pulmonary veins from through the posterior wall. Then one outlet, left anterior anteroventricular or orifice. The blood leaves through the left anteroventricles orifice to the left ventricles. So this is the heart, human being heart structure. Then next slide, the ventricles of the heart. Ventricles are ventricles are discharging chamber of the heart. Discharging chamber of the heart. The pulmonary muscles and tubercules carneae muscles mark ventricular walls. The separated by the intraventricular spectrum, the contains components from the conduction system. The right ventricles pumps blood into the pulmonary trunk. The left ventricles pumps blood into the aorta. The thicker myocardium due to the greater workload, the pulmonary circulation filled by right ventricles is a much low pressure. The system requiring the less energy output by the ventricle. The systematic circulation supplied by the left ventricles by higher pressure system and thus requires a more forceful constructions. So this is the ventricles, the functions, how to the blood discharges from the various cell organs, internal organs. Then next one, the left ventricles. The left ventricles functions mainly. It is wall is a three times so thicker than that the right ventricles one inlet. Left to and anterioventricular orifice one outlet or artific orifice. The two parts were divided by anterior cups of mental value. The inflow tract, rough walls, outflow tract, aortic vestibule, smooth area leading to aortic orifice. Then next one, stepsome fibrous are the skeletons. The internal spectrum located between the right and left atria and continuous force Olis. The intraventricular spectrum, the located between right and left ventricles, upper membranes part, thicker lower muscle parts. The fibrous skeleton, the fibrous ring that surrounded the anteroventricular, pulmonary and aortic orifice, and left and right fibrous regions. This is the anterior view of the human heart, the complete labelings of the anterior heart. The heart valves. The wall is a very important of heart valves. Heart valve ensure the unidirectional blood flow through the heart, composed of an endocardium with the connective tissue core. The two major types. The first one, anteroventricular values and semilunar values. The anteroventricular values, AV values, valves live between the atria and the ventricles. RAV value in tricuspid value, LAV value, bicapsule or mitral value, bicapsule or mitral value. AV valve prevent backflow of blood into the artery when ventricles contact. It is the very important because this art is the blood pumping is the deoxygenated blood one side flow and oxygenated blood to one side flow. So because the values prevent the backflow of ventricles in contact. The chordae tendine, another AV values the papillary muscles of the ventricle valve, prevent the prolapse of the valve back into the atrium. The next one, the semi-lunar heart values. Semi-lunar heart values. This is the function of semi-lunar values prevent the backflow of blood into the ventricles. Have no cardiac tendine attachments. The herotic semilunar value lies between the left values and the and the aorta pulmonary seminar values lies between the right ventricles and pulmonary tongue. The heart sound lub dub due to the values closing. Lub closing the anteroventricular values. The this is the give the sound of the lub. Then closing on the semilunar values automatically the sound is dub. The because the heartbeat sound is the lub dub sound. Then next one, the trapecular values. The God's right anteroventriculars of his three triangular cups, anterior, posterior, and spital. The base of cups are attached to fibrous ring surrounding the anteroventricular orifice. The cardiac tendons find white connective tissue scars attached margin of cups to papillary muscles or mitral valves. God's left to uh, atrioventriculars or face two triangular cups, anterior and posterior, with similar structure to those of right. The value of pulmonary trunk goes to the orifice of pulmonary trunk as three semilunar cups, each with free border. So this is the free here free lobes of three values. So the structure of dissection of three value structure images. Then next one, posterior view. This is the anterior and posterior. Posterior view I'm labeling anatomy of human being heart structure. Then next one, the conducting the system of the heart. Conducting the system. Cardiac muscles tissues has intrinsic ability to generate the conduct the impulse signals. Those cells are contract to rhythmically conduct the system. A series of the specialized cardiac muscle cells, stenoverticals node set the inherited rate of the conductions. Then this is the conducting system of the heart. So this is the vertical node because this is the impact of the nervous systems and nerve functions. Bemman's bundles. This is the bundle of is and arterial nodes, left to posterior bundles and Purkinje fibers. This is the Purkinje fibers here. Then right bundles here, right bundles. Next one, invasion. Innervation heart rate is altered by the external controls. Nerves to the heart includes vesicles, sensory fibers, parasympathetic branches of the vagus nerve, and sympathetic fibers from cervical and upper thoracic chain ganglia. So this is the structural node of SA node, synatural node. The called the pacemaker cells, P cells, the pacemaker cells located at the junction of right atrium and superior vena cave. The upper part of the sulcus terminals upper the epicardium. Then next one, the anterior the anterior ventricle nodes, AV node, atrioventricular. The located in the lower part of the internal spectrum just above the orifice of coronary sinus under the endochondrium. The lower part related to the membranes part of the intraventricular spectrum. Then AV bundles. AV bundles means atrioventricular bundles. This passes forward through the right fibrous region to reach inferior border of membranous part, divides into right and left branches at the upper border of muscular part of the intraventricular spectrum. The next one, major vessels of the heart. The vessels returning blood to the heart includes superior and inferior venae, venae cave open into the right atrium. The return deoxygenated blood from body cells. The coronary sinus opens into the right atrium, returns oxygenated blood from the heart muscles coronary venous. Right and left pulmonary venous open into the left atrium, then returns oxygenated blood from the lungs. The major vesicles for the heart. The vessels conveying blood away from the heart includes pulmonary trunk. The carriers deoxygenated blood from the right ventricles to lungs, splits into the right and left pulmonary arteries. Ascending aorta carries oxygenated blood, the blood way from left to atrium to body organ. The three major branches, bronchocephalic, left common, carotid, left to subclavian artery. So this is the blood flow through the heart. So here, this is the oxygenated blood, this is the deoxygenated blood. So the blood, the pumping is the dimension is lungs to body, body to lungs and heart. So this is the complete oxygen rich. This is the red color is the have the oxygen oxygen blood. This is uh, oxygen poor blood and carbon dioxide rich here. This is the path of the blood through the heart. The next one, disorders of the heart. The coronary heart disease, uh, atherosclerosis fatty despite the arteriosclerosis hardening to the arteries and angina pectoris chest pain, then myocardial infection, blocked coronary artery, silent silent ischemia, no pain or warning, um, fibrillation, irregular heartbeat may occur in the either atrial ventricles. Some disorders of the disease is only genetical disease and some some of the some persons have the particular so this is the image location of the heart left side and so this is the next one, uh, the functioning of heart functions and ECG electrocardiogram, the regulation of the heart rate because of the difference by the nerve impulse and blood clotting, and the next one, uh, Next one, blood. So next one, blood clotting. So blood clotting is a main important of some factors are involved in the blood coagulation. Then blood coagulation is called the blood clotting. The blood outside the vessels quickly changes the from fluid state to thick jelly like materials, which is known as the clot and process of separation of clot from the plasma. The is known as the coagulation or clotting. The blood down from the blood vessels into a test tube changes from the liquid to semi-solid gel in about 4 to 10 minutes of timing of blood clotting timing or blood coagulation timing the outside the vessels of blood clotting time maximum 4 to 10 minutes. The man and other vertebrates and many invertebrates as well. Blood loss is prevented by a series of chemical reaction in which the solid clot is formed to plug the broke vessels. The coagulation or clotting is essential as a formation of plasma, not for the blood cells. This the coagulation function is the only the depends by the plasma. It is not of the blood cells and involves the enzymatic conversion of uh, fibrinogen, a soluble plasma protein, into a fibrin and insoluble protein. This process can be separated under the following steps. Liberation of thromboplastin. Liberation of thromboplastin. It is the release at the time of clotting by the combination of certain factors like phospholipids by repeating the platelets of calcium ions and the anti-hemophilic factors. Anti-hemophilic factors. Then next one, conversion of the prothrombin to thrombin. In this process of calcium ions, the acelerators globin the prothrombin is converted into thro- thrombin by the thromboplastic activity by thromboplastin. The conversion of fibrinogen into fibrin. The proteolytic enzyme thrombin converts the soluble plasma protein fibrinogen into the soluble protein fibrin. The presence of cation and the fibrin stabilizing factor. Those insoluble of fine threads separate from the blood plasma and settle down in the from a fibrous network. The role of blood platelets. The role of blood platelets. Uh, uh, the blood platelets are presumed of to contain the thromboplast. It is released when the blood platelets repeats. The platelets of the blood platelets occurs when the capillaries of blood vessels are damaged and blood flows out. This also lubricates of the number of other factors which activated the process of the clotting or coagulation. Mostly 12 factors are hemaggman factors. Hajman factors is the 12th factors and sixth factors is its presence is denied by the scientist. So all factors are helping to the coagulation of blood clotting. So I show the one slides. So this is the blood clotting pictures observed the when blood is shed out or collected in the container, it loss its fluidity and become a jelly-like mass after few minutes. Like the time in minutes are 6 to 4 minutes or 4 to 10 minutes. This process is called coagulation or clotting of blood. The clot is mass of thin fibrous entailing the blood cells. This fibrous consists of the fibrin. The fibrin is formed from the fibrinogen. See, the factors involved in the blood clotting. The complete 12 factors is here. Coagulation of blood occurs through a series of reactions due to the activation a variety of substances. Those substances necessary for clotting are called clotting factors. The clotting factors are uh, so this is the 12 factors is the first one fibrinogen, second factor prothrombin, third factor thromboplastin, the fourth one calcium, the fifth one labile factor procelerin or accelerator globulin, the seventh one stable factor, eighth one anti-hemophilic factor, and ninth one Christmas factor, 10th one Stewart power factor, 11th one plasma thromboplastin acetylate, and 12th one Hegmer factor, and 13th one fibrin stabilizing factor. So those factors are helping to the coagulation and blood clotting situation. The sequence of the clotting mechanisms. The normally during the circulation, the blood does not clot because the enzymes involved in clotting are in inactivate from slight inside the activation cases clotting in which his enzyme activates. Another one is a uh, sequential manner till the conversion of fibrinogen into fibrin. In general, clotting occurs in three stages is namely mm, formation of prothrombin activator, the conversion of prothrombin into thrombin, and conversion of fibrinogen into fibrin. During the process of blood clotting, the clotting factors which are in activative forms are converted into the active forms, and their enzymatic action produced in the successive reactions are the another in a cascading manner. Thus, Thus, the variation reactions involved in the blood clotting are explained by enzyme cascade theory. So the stage one, the formation of prothrombin activators. The prothrombin activators is formed in two ways, namely extrinsic pathways. In this, the formation of prothrombin activation is initiated by the tissues thromboplastin. The intrinsic pathway in this, the formation of prothrombin activator is initiated by platelets which are within the blood itself. So this is the root map of tissue trauma and tissue thromboplastin protein, glycoprotein, phospholipids. The factor 7th is involved. The factor 9th activated from the 10th calcium factor and 5th thrombin. The prothrombin activator. Then prothrombin activator automatically the tissues are covering the clotting and blood cells. The factors are 12 activated factor 12, the kingdom percolagen and factor 11 activated factor 11, calcium factor 9 they activated from factor 9. Then next one factor 10 activated factor 10, calcium factor V thrombin prothrombin activated. In stage 2 conversion of prothrombin into thrombin. The activator converts prothrombin into thrombin in the presence of calcium. Thrombin itself can accelerate this reaction by positive feedback mechanism. That is the initial thrombin activates factors fifth. This in turn accelerates the formation of both extrinsic and intrinsic prothrombin activator. The next one during this stage, the third stage, The soluble fibrinogen is converted to into insoluble fibrin by thrombonin. Initially, the fibrinogen is covered into the activated fibrinogen, which is called the fibrin monomer. This polymerizes which other manner molecules to the fibrin. The first formed fibrin contains loosely arranged the standards. This is the modified later into a defense tight arrange aggregate by the fibrin stabilizing factors 12. This reaction is requires the presence of calcium ions. The fibrinogen thromboin activated fibrinogen polymerization, fibrin loose standard, and factor 7 the calcium fibrin tight clot. Then anticoagulants. The substance which prevents the uh, postponed coagulation of blood are called anticoagulants. Some substances are used to prevent clotting inside the body in vivo, and some substances used to prevent the clotting outside body in vitro. The variation anticoagulants are the follows: heparin, decomoral, warpin, EDTA, ethynyl aldimate acetic acid, oxalate compounds, ethyl ethyl ND tactic acid. Then citrus, sodium, ammonium or potassium citrate. The anticoagulants are used for this propylaxis, the treatment of thromboembolic disorders for prevention of stored whole blood and blood functions, heparin and acid citrate distors. To keep laboratory blood spectrums from clotting EDTA, citrate, oxalate, and fluoride. The tests for clotting, bleeding time, clotting time, and prothrombin time. The microvasculature. The blood vessels are closed circuits for the transport of blood from the left heart to the metabolic cells. Then back to the right heart, the blood containing oxygen, nutrients, and metabolites is rotated through arteries, arterioles, capillaries, venulus, and veins. These blood vessels differ from each other in the structure and functions. The blood flow from the heart to arteries, which branch and narrow into arterioles. The arterioles give rise to metabolics, which give rise to capillaries. After the tissues has been preferred, capillaries drain into venous, which are then widen more become veins, which return blood to the heart. The micro 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 microarticulation of each organ is specially organized to serve that organ's own special needs, transport of nutrients, the tissues, and removal of circulator extract. The capillary bed is the network of the capillaries supplying an organ. The capillaries are extremely thin structures with walls of the single layer of highly permeable endothelial cells. Hence, interchanges the nutrients and cellular extra occurs between the tissues of the circulating blood. The walls of the arteries of small venous contains relatively large amounts of smooth muscles. The openings of the capillaries are guarded by the mm, muscular pericular spinous. So this is the this is the blood coagulation. blood coagulation, and the functions, factors involved the blood coagulation and factors. How to the reaction, how to the clotting of the blood and internal, external, and time beings. The physically activity classified hard to two kinds of depending upon their mode of contractions, neurogenic and myogenic arts. Based on morphology, those are four types. The sequence of contraction of expressions of cardiac muscles is called heartbeat. The beating is intrinsic character. This stimulus for the contractions of heart starts from arterial node or sinoarticular node. The heart contracts and relaxes at the regular intervals. The contraction is called systole and relaxation is called diastole. A heartbeat includes one systole and one diastole. The heart rate is regulated by extrinsic factors in addition to intrinsic factors. The the former the former factors the former factors are from the nervous and hormonal system. The blood outside the vessels quickly changes from the fluid state to thick jelly-like material, which is known as a clot, and process of separation. Separation of clot from the plasma is known as the coagulation or clotting. The next class will be explain the excretion, the classification of animals, structure and functions of nephron, urine formation, and counter current mechanism, and muscles, types of muscles, ultra structure of skeletal muscles, muscles contraction, and sliding filament theory and energetics, the nerve impulse structure of neuron and nerve impulse synapses. The next class will be taken the physiology part 2 under the excretion, muscles, and nerve impulse. So both three units are next class is explain the next class. So thank you one and all. Please listen clearly your syllabus, and notes will be prepared each and every day. The next class will be going on excretion, muscles, and nervous systems. Thank you one and all.